So in the previous MSc entrance botany question paper, now the next question is the genetic material in TMV is. So TMV, it is a tobacco mosaic virus. It is a plant virus. So we know that all the plant virus contain single strand RNA as a genetic material. And there are few exceptions. And for example, double-stranded RNA is present in the reovirus. That is also plant virus. So it is an exception. In most of the plant viruses, commonly we find single-strand RNA. But reovirus is an exception where we find double-stranded RNA as a genetic material. And single-strand DNA, where we find single-strand DNA? So we find in M13 bacteriophage and also in 5x174 virus. And double-strand DNA, it is present in all the animal viruses and also cauliflower mosaic virus. Now it is also very important. Cauliflower mosaic virus, though it is a plant virus, it is a double-strand DNA virus, cauliflower mosaic virus. So now the answer to the question, the genetic material in TMV, it is very simple question. The answer is single-stranded RNA, but you should remember some of the exceptional cases like the reovirus, which contains double-stranded RNA, and M13 phage and 5x174 virus, which contains single-stranded DNA, and cauliflower mosaic virus, which contains double-stranded DNA as a genetic material. So next, fluid mosaic model was proposed by. So we know there are different models which explain the plasma membrane or the cell membrane. The most popular one is fluid mosaic model. It was proposed by Singer and Nicholson. Okay, and so here you see the structure of the cell membrane. Here the lipid layer, here lipid bilayer, we see the hydrophobic and hydrophilic. The tails, they are hydrophobic, and outer head-like structures, they are hydrophilic. And among these lipid bilayer, the lipid structures, lipid molecules, which show the fluidity. Why it is called fluid mosaic model? Because like in a fluid, the molecule in the fluid, they replace one another, they move. So like that, lipid molecules also, they have the mobility. So it has a fluid nature. And in the lipid membrane, the proteins are arranged in a mosaic pattern. That means irregular pattern. So it is named as fluid mosaic model by Singer and Nicholson. So that is the answer to that. So you see, fluid mosaic model was proposed by Singer and Nicholson. That is the right option. So next question is, the term plasmid was coined by. Who coined the term plasmid? You know, the right option, right answer is Lederberg. Lederberg is the right answer. Okay, and next one is, the double helix DNA model was proposed based on the X-ray diffraction in the studies of. So we know that double helix DNA model was proposed by Crick and Watson, but they proposed this double helical model of the DNA based on the X-ray crystallography structure of the DNA, because there are so many scientists involved initially, like Linus Pauling. Chargaff, Erwin Chargaff, okay, Linus Pauling, he proposed the hydrogen bonds in the nucleic acids, and also Chargaff rule. It is also there related to DNA. Okay, the number of purines equal to number of pyrimidines. That is the Chargaff rule. And also, X-ray crystallographic structure of the DNA was proposed by Franklin and Wilkins. This is also very important. You see, this is the X-ray crystallography model of DNA. Okay, so here you see this is the model. Here you see the pattern of the X-ray diagram. Okay, it shows this kind of structure. So it was interpreted based on this X-ray structure of the DNA. They could they could interpret that it is the helical structure. You see in the diagram, the helical structure of the DNA. Okay, so Wilkin and so this model was proposed by Franklin. Unfortunately, they didn't get the Nobel Prize. Franklin did not get the Nobel Prize, though she contributed a lot. Okay, so and Jacob and Monod, we already discussed that they proposed the lac operon model proposed by Jacob and Monod. Okay, we already discussed about the what is operon. And Frederick Miescher, and he also actually he initially found that there is a structure. He extracted this DNA from the nuclei, and he named it as nuclein. But he didn't find that it is a genetic material. He didn't go further. Okay, Altman, Altman, he worked on mitochondria. Okay, he named them the bioblasts. Altman named the mitochondria as bioblasts. Initially, later it was renamed as mitochondria by Benda. Okay, so you have to remember these names and what are their contributions. Next one, which of the following enzyme is not involved in Krebs cycle? So you see the Krebs cycle here. So here, what are the options given here? Aconitase, decarboxylase, aldolase, and fumarase. Here we have to see aconitase is related to Krebs cycle, decarboxylase. There are different kinds of the decarboxylases in the Krebs cycle. So any decarboxylase we can mention that they are all involved in the Krebs cycle, and fumarase also involved in the Krebs cycle, and aldolase it is involved in the glycolysis or EMP pathway. Just we'll see once. Here you see this is the Krebs cycle, also called as citric acid cycle, because the first product in this cycle is citrate or citric acid. So it is named as citric acid cycle. Also it is named as TCA, tricarboxylic acid cycle, because it contains 3 carboxyl. You see in the structure of the citrate, there are 3 carboxylic groups. So it is also called as tricarboxylic acid, citric acid. So it is named as TCA cycle, tricarboxylic acid cycle. So here you see aconitase. They gave the option aconitase. It is involved in the Krebs cycle. And dehydrogenases, and decarboxylases. Also, because here isocitrate decarboxylase also involved here, because carbon dioxide is released in this reaction. And fumarase also involved in this Krebs cycle. And if you see the glycolysis, in the glycolysis you see the glucose is a substrate here, and hexokinase, phosphoglucose, isomerase, and phosphofructokinase. And next here we see the aldolase. Aldolase involved the reaction here, fructose 1,6-bisphosphate, it is cleaved into two compounds. Fructose 1,6-bisphosphate is a 6-carbon compound. It is broken or cleaved into two compounds which are 3-carbon compounds. Glycerol head 3-phosphate and dihydroxyacetone phosphate, DHAP. Okay, and so aldolase is involved in the glycolysis, whereas the three remaining enzymes are involved in the Krebs cycle given in the question. So you see, Krebs, which of the following enzyme is not involved in Krebs cycle? So aconitase, decarboxylase, and fumarase, they are involved in the Krebs cycle, whereas aldolase is clearly it is involved in the glycolysis. So the right option is aldolase. It is not involved. Next, male gametophyte of pinus are, is it 2-cell, 4-cell, 6-cell, 10-cell? So in case of the angiosperms, we see the male gametophyte. Finally, at the time of pollination, it is 2-celled. But pinus, being a gymnosperm, here, if you see the diagram here, pinus, this is the, we already discussed the male gametophyte or the pollen grain or the microspore, which contain two wings. Winged pollen grains is the important feature of pinus. So we see the wind-mediated pollination here. Okay, anemophily, it is called as anemophily. That is an important feature of the pollen grains in the pinus. They are winged. There are two wings present here. And here you see in the male gametophyte, the first prothallel cell is there, and antheridial cell is there, and first prothallel cell undergoes one more division. It forms second prothallel cell. First and second prothallel cell is the antheridial cell. Next, it undergoes division to form generative cell and tube nucleus. So finally, at the time of the pollination, means at the mature male gametophyte, before pollination, we see 4 cell stages: 2 prothallel cells, generative cell, and tube nucleus. So the right answer is 4-celled. Male gametophytes of pinus are 4-celled at the mature stage. So that is the right answer. And next, one of the following components are absent in the core of bacterial spore. So we know the asexual reproduction of the bacteria. We see the spore formation. Under the favorable conditions, the bacteria undergo binary fission. But under the unfavorable conditions, bacteria form the spores. They are also called as endospores, which consists of ribosomes, amino acids, and DNA. But there is no tRNA in the spores of the bacteria, in the endospores. So the right answer is one of the following components are absent in the core of bacterial spore. Here, tRNA. tRNA is not present in the, it is not found in the spores of the bacteria. Next, whiskey and rum are produced from the molasses of the family members of. So these whiskey and rum, these are the, actually in the sugar industry, molasses is the byproduct. From the molasses, alcohol is manufactured, and that, and also useful for the making beverages like whiskey and rum. So they are made from the barley. Whiskey is made from the sugar cane. Okay, it is a, in the sugarcane industry, while making the sugar, that molasses is the byproduct. From that, whiskey is made. Okay, from the barley, after the distillation, rum is made. So these are the products of the distillation of the molasses. So from where this barley, sugarcane, all these are the plants which come under Poaceae or Graminae. Okay, so the right answer is Poaceae. They belong to the Poaceae. All these sugarcane or barley, all these. Next, single strand DNA is the genome in. Okay, single stranded DNA, already we discussed about this. What are the genetic materials which are present in TMV? In TMV, it is a single strand RNA, not DNA. SARS virus, it's a retrovirus. Okay, it is a double. This virus consists of RNA as a genetic material. Okay, SARS virus, it contains RNA. Coronavirus, all of them belong to same family. They cause the respiratory diseases in the human beings. And HIV, it's a retrovirus. It is also RNA virus. And the question is about the single strand DNA. We already discussed that single strand DNA is present in 5 into 174 virus and also M13 phos. It is not given here. Single strand DNA is present in M13 phos also. So particularly M13 phos is used in the cloning experiments also, since it contains single strand DNA. So the right answer is this fourth one. Next, chemiosmosis theory was proposed by. So who proposed? What is the chemiosmosis theory? So once we'll see. So this here, chemiosmosis theory is related to ATP synthesis. Synthesis of ATP from ADP and hydrogen ion, that they are also called protons. So in case of the oxidative phosphorylation or the photophosphorylation, what happens? The protons which are accumulated in the membrane between the two membrane structures. Okay, due to the proton gradient, that is also called as proton motive force. Proton, these are protons, not proteins. H plus ions are called as protons because they don't have neutrons, they don't have electrons, they don't only have the protons. So they are H plus ions also called as protons. So in the electron transport, during the electron transport, what happens? The H plus ions, they move into the membrane, between the two membranes, and they form the gradient. Okay, so they form the proton motive force, which force the H plus ions into the lacuna inside. At that time, see, protons are moving through the ATP synthase. So ATP synthase is present in the inner membrane. This is the outer membrane and this is the inner membrane. In the inner membrane, ATP synthase is present. It is useful in the synthesis of the ATP from ADP plus inorganic phosphate. While hydrogen ions or protons are moving through the ATP synthase due to the gradient difference, that is called as proton motive force. So this is the theory. Based on this, they propose the chemiosmotic theory, chemiosmosis. Peter Michel proposed this theory and he got the Nobel Prize for this. So that is the answer for this. Peter Michel proposed this chemiosmosis theory and he got the Nobel Prizes for this. It is related to ATP synthesis. ATP synthesis, also called as phosphorylation. There are two types of the photophosphorylation. In the chloroplast, we observe the photophosphorylation in the light phase, and we find the oxidative phosphorylation in the mitochondria during the electron transport. In both the cases, ATP is synthesized with the help of the ATP synthase. So it was explained with the help of the chemiosmosis theory by Peter Michel. Next, double fertilization involves. So here you see double fertilization You see the double during the double fertilization, how the double fertilization occurs. You see here in the embryo sac or female gametophyte, we see one egg cell and two synergids. Okay, during at the side of a micropylar end, and at the center there are two polar nuclei or central cell, and at the chelagel we find the three antipodal cells. Now what happens after the pollination? After the pollination, pollen grain, it starts germination. It forms the pollen tube. So you see here in this picture, after the pollination, see pollen grain, after pollination, it starts producing the pollen tube and it enters into the embryo sac, or also called as female gametophyte, and it carries two male nuclei or male gametes. You see in this picture. So the generative cell, it undergoes division and it forms two male gametes. Okay, tube nucleus, it degenerates. Say two male gametes are produced here, or these are also called as sperm cells. And here you see how they enter into the embryo sac through the pollen tube. The synergids, they degenerate. One of the synergids degenerates and two male nuclei are sperm nuclei. See, one of them fertilizes the X nucleus, and second one fertilizes the polar nuclei which are present in the central cell. So this process is called as double fertilization. So the product of the fertilization with the egg cell is zygote, whereas the product of the triple fusion or double fertilization is 3N cell, that is the PEN, primary endosperm nucleus, that is the triploid one. So that is the one. Here you see, so in double fertilization involves, what it involves? Fertilization of the egg by two male gametes. Actually, one male gamete only. Fertilization of the egg by one first male gamete only, not two male gametes. That is a wrong option. And fertilization of two eggs in the same embryo sac by two male gametes. Actually, in egg apparatus there is only one egg cell, so it is also a wrong option. Fertilization of the egg and the central cell by two male gametes brought by two different pollen tubes. Actually, the fertilization occurs separately. Fertilization of the egg and the central cell by two male gametes, that is true, but brought by two different pollen tubes is wrong. They are brought by a single pollen tube, the same pollen tube. That is the right answer here. Fertilization of the egg and the central cell by two male gametes brought by the same pollen tube. So that is the right option here. And next, which enzyme complex is responsible for the reduction of molecular nitrogen to the level of ammonia in root nodule of legumes? So we know about the nitrogen fixation. In the nitrogen fixation, what happens? Nitrogen, the gaseous nitrogen cannot be utilized by the plants, so it should be converted, okay, into ammonia. So it is called as nitrogen fixation. So here the enzyme which is involved, what is the enzyme involved in the legumes? You see in the rhizobium, you see the rhizobium, it is a symbiotic nitrogen fixing bacteria, and there are also other non-symbiotic bacteria also which are involved in the nitrogen fixation. But with reference to legumes in the root nodules, the rhizobium consists of the one enzyme that is called as nitrogenase. The right answer is nitrogenase. So here you see nitrogenase enzyme. It is a complex which consists of dinitrogenase reductase and dinitrogenase. Both are involved in the nitrogen fixation. And this nitrogenase enzyme complex is inactivated by the presence of the oxygen. So that's why there is one pigment called leg hemoglobin, which is present in the nodules, root nodules, because it readily binds the oxygen. This leg hemoglobin has affinity to bind the oxygen molecule. So it prevents the oxygen to enter into the inside the legume root nodule cells. Because if the oxygen enters, because plants, they carry on respiration from the in the respiration, oxygen is released, but due to the presence of the leg hemoglobin, it acts as a wall. It binds to the oxygen which is released. So nitrogen is cannot be inactivated. So here the which enzyme complex is responsible for the reduction of the molecular nitrogen to level of ammonia in root nodule of legume? The right answer is nitrogenase, and the pigment which is present in the root nodules is leg hemoglobin. That also should remember. And also there are some genes which are involved in the nitrogen fixation. They are called as NIF genes, NIF. So that also you should remember. Next, quantasomes are found in. What are the quantasomes? We already discussed that quantasomes are the functional and structural units of the photosynthesis which are present in the chloroplast. Particularly, they are present in the thylakoid membranes of the chloroplast. And you see, so quantasomes, you see the thylakoids which are present in the chloroplast, in the thylakoid membranes, all the pigments, lipids, proteins, they form a complex structure that is called as quantasome. These are called as structural and functional unit of the photosynthesis. So that is the right answer. Quantasomes are found in, not in mitochondria, not in nucleus, not in lysosome. They are found in thylakoid membranes of the chloroplast. Okay, that is the right answer. And next, angular leaf spot of cotton is caused by. What is the pathogen which causes angular leaf spot in cotton? The right answer is Xanthomonas. And that is the full name is Xanthomonas malvacearum. Okay, and Colletotrichum, it causes the rot disease in sugarcane, Colletotrichum falcatum. Okay, and also Pyriclaria, it causes blast disease in rice or paddy. Okay, rice blast, Pyriclaria vareza. So the right answer here is angular leaf spot of cotton is caused by Xanthomonas malvacearum. And you know, these diseases are very important. Already we discussed citrus canker, also caused by Xanthomonas oxynopodis citric, that we already discussed. Xanthomonas citric, we can also call it as. Okay, and a blight disease of the paddy or rice is caused by Xanthomonas varese. So they may ask, particularly Xanthomonas malvacearum causes angular leaf spot disease or knot, black knot disease. This is also called as black knot disease in cotton. Xanthomonas malvacearum. So you should remember all the pathogen names, particularly Xanthomonas malvacearum, Xanthomonas oxynopodis citric, Xanthomonas varese. What are the diseases they cause? Next, conidiophore is 2,2,4-septating. Conidiophore. So the answers given are Specilotheca, Fusarium, Colletotrichum, and Pyriclaria. Pyriclaria, actually, Pyriclaria is related to our syllabus, so we can give the answer Pyriclaria, but the Fusarium also right answer here. They gave both the answers. Fusarium is not part of our syllabus, but Fusarium conidiophore also contains 2 to 4 septa. Okay, but the nearest answer is pyraclaria here, since it is in part of our syllabus. And next, in angiosperms, tunica carpus organization is seen in. So there are some theories proposed to explain the shoot apex structure and root apex structure, and apical cell theory, histogen theory. So they are Carper Kappe theory. They are related to root apex, and this apical cell theory and tunica carpus theory, Newman's theory, French concept. All these are related to shoot apex. So here we already discussed this tunica carpus organization is related to shoot apex. So that is the right answer. Ornstein, Schmidt, all these names you should remember. Schmidt, who proposed this tunica carpus theory, who proposed histogen theory, who proposed apical cell theory, Nageli, Einstein. So all these, Nageli, you should remember, Hofmeister, Nageli and Hofmeister are related to apical cell theory, Einstein related to histogen theory, and Schmidt related to tunica carpus theory. So you remember all these names. Next, Beadle and Tatum identified specific mutations. So they, Beadle and Tatum, they worked on some mutations, specific mutations. Okay, auxotrophs, like how the nutrient deficient, they created some of the nutrient deficient varieties by mutation. Okay, the so that they use the Neurospora crassa, that is a fungal organum, Neurospora crassa. They used this organum, fungal organum, for their studies on the mutation specific. They created some specific mutations in Neurospora crassa, which are nutrient deficient varieties. Beadle and Tata. And also they proposed one theory, one gene, one enzyme theory, one enzyme. That also you should remember. Beadle and Tatum, they proposed one famous theory that is called as one gene, and one enzyme. That means one gene is responsible for the synthesis of one enzyme. But later it was modified as one gene, one polypeptide theory. But you should remember, Beadle and Tatum were related to their mutations in the Neurospora crassa and also one gene, one enzyme theory. Next, sargassum reproduces vegetatively. Actually, sargassum is not part of our syllabus, but since they gave the fragmentation, vegetative reproduction occurs through the fragmentation. That means their algal filaments, they are fragmented, they are cut into small fragments. Each fragment can grow independently as a full algae. So that is called as vegetative reproduction by fragmentation in the sargassum. Next, apomixis is, so it is related to embryology. Okay, polyembryony, apomixis, these are the topics there. Here, apomixis is related to the formation of the seeds without any sexual reproduction, without any meiosis. Okay, that you should remember. Apomixis is the formation of the seeds. The fruit is formed, seed is formed, but without sexual reproduction or meiosis. This is very peculiar process. So the right answer, the formation of seeds by fusion of male and female gametes, it is wrong. That means there is no fertilization at all. If there is fertilization, it is not called apomixis. Formation of seeds with syngamy but not meiosis. That is also not true, because syngamy involves fertilization. And formation of seed without syngamy and meiosis, that is the right answer. How it happens? How it happens? See, during the formation of the, we also know about the megasporogenesis. Megasporogenesis means what? Megaspore mother cell, which undergoes meiosis. In usual cases, normal cases, megaspore mother cell in the new cellus of the ovule, it undergoes meiosis and it forms 4 haploid megaspores. Out of the 4 haploid megaspores, one degenerates. Okay, 1 megaspore, 3 megaspores degenerate, and one remains as the functional megaspore, and it undergoes 3 successive mitotic divisions to form the female gametophyte with the 8 nuclei. That we know. But in some cases, what happens? This diploid megaspore mother cell, instead of undergoing meiosis, it directly forms a diploid embryo sac or diploid female gametophyte. Sometimes it directly forms the embryo. Directly it forms the embryo. So when the diploid megaspore mother cell, it directly forms the embryo. That is also called as diplospory. It forms a diploid embryo without fertilization and without meiosis. And sometimes you may study it in the polyembryony. Sometimes integuments also form the embryos. Integuments of the ovule. And from the nucellus also sometimes embryos are formed. Sorry. So here in the apomixis, what happens? Seeds are formed with the embryos without syngamy and without meiosis. The right answer is this third option. The next one is the reaction center in photosystem 2 has an absorption maximoid. So there are 2 photosystems involved, photosystem 1, photosystem 2 in the light phase. Okay, we already discussed in the thylakoid membrane. These 2 form, photosystem 1 and photosystem 2 are present, and they have reaction centers. The reaction center of PS1 is called as P700. The reaction center of PS2 is called as P680. Why it is called P680? Because it has the absorption maxima of 680 nanometers. Okay, it absorbs the light with a wavelength of 680 nanometers. So that reaction center is called as P680 in the photosystem 2. That you should remember. Okay, and here fungus known by the name of bread mold. The answer is Rhizopus. We know it. Rhizopus is called as black mold or bread mold.